everyone, my name is Korawich buasri. Now I will talk about the implanted gene targeting can be enhanced by the use of CRISPR-Cas12a.
First, I will talk about the gene targeting. The gene targeting or GT is an in vivo genetic modification technique that uses homologous recombination to modify gene target. And this process needs a DNA editing template for repair. This figure shows the mechanism of repairing when have a DNA double strand break. The gene targeting or GT can be achieved by the induction of double strand break with a specific sequence endonuclease. When the DNA break, DNA will repair itself, which there are two ways for repair. The first way is HR. The HR is homologous recombination, and the second way is NHEJ. The NHEJ is non-homologous enjoining. The difference between HR and NHEJ is HR usually used for gene modifier that can control base change, which require for GT, because GT require HR as a DNA repair, and then HR still need a DNA editing template for repair. But the NHEJ usually used for gene knockout that can't control base change, which is a need for GT, and it don't need a DNA editing template for repair, which different from HR. So the NHEJ does not need for GT. The problem is HR have a low level in plant cell because generally the somatic cell of plant usually occur the NHEJ more than HR. So this problem is cause a low GT efficiency. In previous study of them, they can improve the GT efficiency about 1%, but it's not satisfying. So they want to find a way that can improve the GT efficiency. The objective of this study, the first objective, to test the absence of HR suppressor can improve the GT efficiency, and the second objective to compare the GT efficiency between LB-Cas12a and SA-Cas9. The outline in this study, they have two aims. The first aim to compare the GT efficiency between present and absence of HR suppressor, and the second aim to compare the GT efficiency between the use of SA-Cas9 and LB-Cas12a. The first outline, they compare the present and absence of HR suppressor to check the absence of HR suppressor can improve the GT efficiency or not. In previous study told them that the HR have a suppressor which have a RTEL1, IMI2, and the last one is FANCM1. If HR was inhibited, so the GT efficiency may be reduced because GT use HR for modifying gene. So they are interested in HR suppressor because they want to know if have a absence of HR suppressor, it can improve the GT efficiency or not. So they use the mutant line that have a two mutation of gene in each mutant. The first mutant have a mutation of RTEL1, IMI2, and the second mutant have a mutation of RTEL1 and FANCM1. Next, they create the DNA construct of ALS gene. The DNA construct contains a Cas gene, Cas9 gene. The Cas9 gene that they use in this study is a SA-Cas9 because it provides the most efficiency from previous study and use egg cell specific promoter for expression and contain a gRNA, contain a HR donor. The HR donor has a point mutation. And the last one contain a PPT resistant. The PPT resistant is a selectable marker, and the gene target is a ALS gene. That they choose the ALS gene in this study because if ALS gene has a point mutation, it can resistant to specific herbicide. When they succeed in creating the tRNA construct, they transform the vector that contain the tRNA construct into a Arabidopsis plant. When the tRNA construct integrate with the Arabidopsis genome, and then if Cas9 gene and gRNA have a expression, the product of gRNA will bind with Cas protein and take it to target sequence and lead to induced DNA double-strand break on recognition site. After the DNA target is cut, it will repair by HR with HR donor. Finally, they get the tRNA construct that have a point mutation in ALS locus in Arabidopsis plant, which it can resistant to specific herbicide. The herbicide in this study is a imazapyl. Next, they transform the vector that contain the tRNA construct into Arabidopsis plant by follow dipping technique. To select the transgenic plant, they use the PPT for select the transgenic plant. If have a tRNA construct transformed in Arabidopsis, this plant it can resistant to PPT. The plant that can resistant to PPT is a T1 line. And then they harvest the seed of T1 line and plant on medium that contain a imazapyl. If have a GT event, this plant it can grow. Then they calculate the GT efficiency of each individual of T and calculate the mean GT rate to compare the GT efficiency between each approach. This graph show comparing of GT efficiency between wild type and mutant. The results show that the wild type give a higher GT efficiency more than mutant. In this table, I want you to focus on the mean GT rate, which will tell the percentage of GT. It can be seen that the mean GT rate of wild type is higher more than the mean GT rate of mutant. So, the absence of HR suppressor does not enhance the GT efficiency. Next, the second outline, they compare the use of LB-Cas12a and the use of SA-Cas9 to check the use of LB-Cas12a can improve the GT efficiency or not. This figure shows the comparing the cutting pattern between CRISPR-Cas9 and CRISPR-Cas12a. The CRISPR-Cas9 is cut the DNA target into a branch end, which the repairing of branch cutting pattern usually occur and HEJ more than HR, while the CRISPR-Cas12a is cut the DNA target into a sticky end, which the repelling of sticky end cutting pattern usually occur the HR more than NHEJ. So the use of LB-Cas12a may be improve the GT efficiency. Next, they create the gene a construct of ALS gene for use with LB-Cas12a. This construct is similar to the gene a construct of Cas9 but different as site of Cas gene and gRNA. This graph shows the interrelate between the LB-Cas12a, SX-Cas9 and untransformed. That they use the interrelate in this study because interrelate tell about the rate of deletion and insertion which repair almost all by NHEJ. So if the interrelate low, show that the NHEJ occur a little. The result of this graph is the interrelate of LB-Cas12a is lower than the interrelate of SX-Cas9. So the use of LB-Cas12a give a HR higher more than the SX-Cas9. And then the use of LB-Cas12a may be improve the GT efficiency. Next, they check the efficiency of the use LB-Cas12a and SX-Cas9 in increase the GT efficiency. This figure shows comparing of GT efficiency between LB-Cas12a and SX-Cas9. The results show that the use of LB-Cas12a give a higher GT efficiency more than the use of SX-Cas9 because the average of LB-Cas12a higher than of SX-Cas9. In this table, I want you to focus on the mean GT rate, which will tell the percentage of GT. It can be seen that the mean GT rate of LB-Cas12a is higher more than the mean GT rate of SX-Cas9. So the use of LB-Cas12a is better than the use of SX-Cas9. The conclusion. The first conclusion from the objective to test the absence of HR suppressor can improve the GT efficiency. Conclude the absence of HR suppressor does not enhance the GT efficiency. The second conclude from to compare the GT efficiency between LB-Cas12a and SX-Cas9. Conclude the use of LB-Cas12a was superior over the use of SX-Cas9. I would like to say thank you to Ajahn Supachai Wutipong Chaikit for supporting and giving an advice. Thank you for your attention.
If you have any question, you can ask me via email.